Good morning, good afternoon, and good evening, everyone. And welcome to the Scientist.com and Xenotech webinar titled Are In Vitro Metabolism and Drug-Drug Interaction Studies Critical for an Investigational New Drug Application? I'm Liam Sanyo from the events team here at Scientist.com, and I'll be your host today. We're being joined by Dr. Andrew Taylor, Manager of Technical Support for Services at Xenotech, who will provide an overview of the processes by which drugs are metabolized and discuss the importance of drug metabolism and drug-drug interaction studies. And with that, I'd like to welcome Dr. Andrew Taylor. Thanks so much for joining us today, Andrew, and the floor is yours whenever you're ready.
Thank you very much for that great introduction, and good morning, good afternoon, good evening to all of you joining us, depending on where you are in the world. Thank you for taking the time. Today, I'm going to be presenting on are in vitro metabolism and drug-drug interaction studies critical for an IND submission. Just a brief overview of what the session and the objectives are. So drug metabolism and drug-drug interaction studies often take a backseat to toxicity studies when the drug developer is preparing their IND. However, benefits to drug development efficiency and expense, along with modern regulatory expectations, are causing many to look deeper much earlier on in the process. So the question comes up often, when is it most beneficial to run these studies, and which of these studies should be run? So some of the goals that I hope to accomplish are, again, talking about why run ADME DDI studies, when to conduct these studies, and some areas of concern that can be related to the studies. And that has to do with proper design and interpretation of the data. So let's get into it. Are in vitro metabolism and DDI studies critical for an IND? So just to give a brief outline of the presentation today, I'm first going to talk about why a drug developer would run these studies. Then we'll take a look at the predominant types of in vitro ADME and drug-drug interaction studies that are available to the drug developer. We'll talk about the importance of proper study design and data interpretation by looking at a few case studies. And then we'll follow up with a discussion on when to conduct in vitro ADME DDI studies and the timing of these studies in the drug development process. And lastly, we'll wrap up just looking at some additional highlights and information from today's talk. So first, the main question, why conduct these studies? Is this just a box checking exercise? And that's a question that we get asked a lot by clients and sponsors. And I'm just going to start out and give the answer right up front that although a lot of sponsors are under the impression that these are just box checking exercises, the answer is an emphatic no. These are not just box checking exercises. These studies are pivotal in providing information on multiple important aspects of the drug. First of all, these studies provide a deeper understanding of the drug molecule itself, things such as its metabolism, the enzymes involved in its metabolism, and so on. But in addition to that, if you look at the bigger picture, the main purpose of these studies are to inform the patients and the healthcare professionals about the safe polypharmacy, which is a common practice to manage multiple health conditions. Most of the information generated from these drug-drug interaction ADME studies is used during the drug label negotiations, and ultimately, the drug label guides the physician and the pharmacist to determine which drugs can or cannot be given concomitantly, or in certain situations, perhaps the dose of the drug needs to be adjusted in such a way that there are no undesired effects from taking these drugs in combination. An example that can be given here is of the drug warfarin, which is an anticoagulant drug, and it has a very narrow therapeutic index. So warfarin is predominantly metabolized by CYP2C9. So if there is a drug that induces CYP2C9, and this is given alongside a warfarin, it's going to increase the clearance of warfarin, thereby decreasing the efficacy of warfarin and this could lead to the likelihood of clot formation in patients. So therefore, knowing the potential drug-drug interaction is of immense importance. Additionally, from the pharma company's perspective, these studies can help to decide on different drug candidates early on in the drug discovery process. So if there's a drug candidate that has potential considerable drug-drug interaction liability, this is known up front, thus saving potential time and money. Lastly, the results from these studies also help to determine the first-in-human dose and the need for any clinical DDI studies. And also, really importantly, yes, at the end of the day, these studies are box-checking exercises, and the data obtained from them do furnish the required information for the drug filing as per the regulatory guidance requirements. So the takeaway message, even at the beginning of this talk, is that while these studies may appear to be deceptively simple, maximizing the amount of insight that can be gained from them can be complex, but is a critical step in the drug development process. So there are three main guidance documents available that relate to in vitro drug-drug interaction studies. These include the FDA's guidance for industry, with the final version that was put out in January of 2020. The European Union, the EMA guidance, the final guidance was put out in 2013. It's currently undergoing some revision. And Japan's PMDA guidance, with the final version that was put out in 2019. Now, one thing I'd just like to briefly note about these various guidances is that over the years, there's been somewhat of a harmonization between what these guidances are asking from industry. There are, of course, some caveats and nuances between them. And to get a good rundown of those, I direct you to our website, where Dr. Brian Ogilvie has put out some very good information comparing these different guidance documents, where they're similar and where they differ. So in addition to those three guidance documents, there are also two other guidance documents that talk about the necessity of PK and metabolite characterization and safety studies. The first one of these is the safety testing of drug metabolites, or the MIST guidance, which is in its second revision. And the other one of these is the M3R2 guidance for industry, which also talks about some of these aspects. And this one, I'm sure a lot of the toxicologists are a lot more familiar with. So ADME and drug-drug interactions. So ADME, which stands for the absorption, distribution, metabolism, and excretion, these are predominantly driven by two cellular elements that can be altered. The first of these are drug transporters. And the drug transporters drive the absorption, distribution, and excretion of the drug. The second of these cellular processes are the drug metabolizing enzymes that metabolize or clear the drugs from systemic circulation. So as you can see here in the simplified scheme, that if one drug affects either the transporters or the drug metabolizing enzymes, or both, it can impact the overall ADME of the other drug and can result in undesired effects, such as a decrease in efficacy due to decreased exposure, or perhaps an increase in the efficacy of the drug, or even toxicity due to increased exposure of the drug. Now, some of these effects can be caused by either exposure to the parent drug, or by an increased metabolism of the parent drug into toxic metabolites. So in order to address a lot of these potential impacts that can occur with drug-drug interactions affecting the ADME profile of a drug, there are various types of in vitro studies that determine the ADME and the DDI potential of a drug. And they're shown here in this chart, and they can be broadly grouped into three subsets. The first category is drug metabolism, and this is driving the M and the E portions of ADME and is characterized in three separate study types. The first study type in this category is the assessment of a comparative metabolism across various species. The second study identifies different metabolites that are formed from the parent drug and how these various metabolites can compare across various species. And the third metabolism type of study is called a reaction phenotyping study. And this is typically conducted to evaluate the relative contribution of various CYP enzymes in the metabolism of the drug. In the second category, the drug is evaluated for its potential for inhibition or induction of various drug metabolizing enzymes. These studies include CYP inhibition, again, where you're profiling for, is the drug a specific inhibitor to various CYP isoforms? Or CYP induction. What is the induction potential of the drug for given CYP isoforms? The third category of studies is the drug transporters. And this is looking at the substrate or inhibition potential of the drug for various drug transporters. Now, I'm going to go into these study types in more detail here coming up. But one thing I would like to mention here is these studies do not necessarily need to be run in the order that's shown here. And we'll discuss that a little bit more at the very end of the presentation too, when we talk about the timing of in vitro ADME DDI studies. And lastly, before moving on, I'd also just like to very briefly mention the test systems that are available for in vitro drug-drug interaction experiments. And again, we'll talk on test systems when we talk about some case studies. But selection of the appropriate test system for these studies is of great importance when designing these study types. So various types of in vitro studies that are used to determine the ADME and DDI potential of a drug have been shown. And again, all of these different experiments can be done in different test systems. Some of these test systems include cryopreserved primary hepatocytes. And primary hepatocytes are kind of considered the gold standard when it comes to drug metabolism studies. Hepatocytes are intact living cells that are isolated directly from living liver tissue and frozen for use at a later time. Because these are intact cells from living tissues, they have all the metabolic pathways functioning within them, and they don't require the addition of any additional cofactors to activate some of these various drug metabolizing enzymes. However, there are different situations where other test systems might be a better choice for use. And some of these other test systems include things that we call subcellular fractions. And subcellular fractions are non-living cell tissue lysates that have been fractionated to enrich for certain enzyme activities. And these are generally made from either liver tissue or other tissues as needed. And these include subcellular fractions such as S9, microsomes, and cytosol. Now, it's beyond the scope of today's talk to go into a lot of the details between these different test systems, but just keep in mind that the selection of the most appropriate test system is very important when designing your in vitro drug-drug interaction study. So let's start by reviewing these studies, first starting with drug metabolism studies. So the first study type is we'll talk about interspecies comparative metabolism. And this is a quick metabolic stability screening study that's usually performed across various species. The test system chosen for this can be hepatocytes or some sort of other subcellular fraction, again, such as microsomes or S9, again, from various species comparing to human. Now, this study is a very useful tool for drug developers and is used to determine which animal species might be the best to choose for IND-enabling tox studies when looking for the animal species that is most human relevant. So, for example, here, if we take a look at these plots, all the plots are the same. The percent remaining drug is shown on the x-axis as a function of time on the y-axis. So we're incubating the drug, in this case in hepatocytes, and looking at its clearance over time. And as you can see here in this case, we're looking at rat, dog, monkey, and human. Now, these results show that human and monkey have about the most similar metabolic profile when it comes to the clearance of the drug, where the clearance of the drug is much more rapid in the dog and much more slower in the rat. So overall, just looking at metabolic clearance, in this case, monkey would be the most reasonable choice for a non-clinical tox species. This study is also useful during lead optimization phase when there could be several compounds in the pipeline and the drug developer is trying to down select some potential candidates. Again, trying to find the most proper candidate to use to move forward to the next level. So the next study type when it comes to looking at drug metabolism would be an interspecies comparative metabolite ID study. Now the goal of this study type is to determine the various metabolites that are formed from the parent drug in various species and to figure out which of those species are most similar to human. And this type of study also helps the drug developer identify any human specific or human disproportionate metabolites that may be being formed, or also if there are any toxic metabolites that might happen to be formed. Now, both of these things are all very important for the drug developer because you want to be able to select a species that is similar in metabolism to human. And you also want to know early on if there are any human-specific metabolites for which the drug developer might need to conduct independent tox studies for down the road, or metabolites, abundant metabolites that might need some additional in vitro ADME work done on them themselves. So just here is an example showing the drug coumarin. Now in humans, coumarin is converted to 7-hydroxycoumarin, which is a non-toxic metabolite. However, in rats, coumarin is metabolized to an epoxide, which is very hepatotoxic. So again, this study type is of particular interest to the drug developers and toxicologists because it provides the information for species selection for non-clinical tox studies and is also telling if there are any human-specific metabolites or disproportionate human metabolites or toxic human metabolites very early on, and again, prior to a clinical study when the drug is introduced into humans. Again, just sticking on the subject of metabolite ID. So LC, Tanaman analysis identifies the various metabolites, which is shown here in this example. So on the right side is a product ion spectra of one of the metabolites. Now, one thing to be aware of when looking at these data is that for the most part, these data give a qualitative profile of the metabolites that are formed from the drug. And this is because the peak height that is that is measured is determined by the ionization efficiency of the various metabolites, and it's not necessarily related to the quantity of metabolites that are present. So in order to get a quantitative assessment, the inclusion of metabolite reference standards needs to be done, or the compound needs to be radio labeled so it can be detected radiometrically while also looking at it in the mass spec. There's one sort of middle ground, and that is metabolites that are absorbed in the UV spectra. And if there are any metabolites that happen to be absorbed in the UV spectra, these can be better compared in a semi-quantitative manner. There's a lot to say on this subject, and again, if you're interested in learning more about this in a lot more detail, again, I would like to direct you to our website where we have some great videos that discuss this topic in much greater depth. Lastly, this table here provides an example of the type of data that can be obtained from a cross-species MetID study. So the various rows are the components or metabolites that were identified, and the various columns are different pieces of information, such as the mass shift of the metabolite, which allows you to come up with a proposed biotransformation that happened to the parent molecule, and also if this metabolite was found in these various species, mouse, rat, dog, pig, or human. So in this example, the components or metabolites 1 through 5 are formed in all the species that are tested. However, you can see here in the bottom circled, component 6 is only formed in humans and pigs. And that indicates for this example that pig would be the better choice for the tox studies going forward, as opposed to the other species, as it's the most similar to human. The next and last study type when it comes to drug metabolism in vitro ADME studies is reaction phenotyping. Reaction phenotyping is used to identify the relative contribution of various CYP enzymes that are involved in the metabolism of a drug. In this study design, the drug is incubated with recombinant CYP enzymes, and either the formation of a given metabolite, the loss of the parent, or both are measured, and the rate of metabolism is plotted. The data obtained from the recombinant incubation with the recombinant enzymes is corroborated by incubating the drug in human liver microsomes as well, in the presence and absence of CYP inhibitors that are specific to each CYP isoform. And again, the loss of the parent or formation of a given metabolite is monitored. So the data obtained by these two experiments, both the recombinant CYP enzyme incubation and incubation in human liver microsomes using selective inhibitors, complement one another. And in combination, these two data sets provide a picture of the metabolic pathways involved in the drug metabolism. Now, additional studies looking at non-CYP enzymes, such as UGTs or carboxylesterases, can also be performed in human liver microsomes or evaluated in hepatocytes. So as shown in this example here, the drug being tested is terfenadine. Now, terfenadine is a prodrug, and it's rapidly converted to its active form, fexofenadine, by CYP3A4. So terfenadine was the first non-drowsy daily antihistaminic drug, and it was brought to market in 1985. When people were taking this drug alone for seasonal allergies, it was completely safe. However, if a person happened to develop a bacterial infection and at the same time was given an antibiotic such as erythromycin or some other sort of macrolide antibiotic as a treatment, in some cases this turned out to be a fatal drug-drug interaction. Now, the mechanism behind this was because erythromycin is an inhibitor of CYP3A4. So by inhibiting CYP3A4, which is the main enzyme responsible for the metabolism of terfenadine, it can lead to an increased systemic exposure of terfenadine, which in turn can cause QT prolongation and death. So because of this, terfenadine was pulled from the market due to this dangerous drug-drug interaction with CYP3A4 inhibitor drugs. Now, Allegra, or fexofenadine, which is a metabolite of terfenadine, turned out to be a much safer alternative and is still a drug that's on the market today. So you can see here in this plot that it's showing the rate of metabolism on the y-axis as a function of the CYP isoforms on the x-axis. And again, in this case, it's examined using recombinant human CYP enzymes. So you can see here, it's pretty clear that the conversion of terfenadine to fexofenadine is predominantly catalyzed by CYP3A4. So therefore, this shows how taking terfenadine in conjunction with a CYP3A4 inhibitor can lead to the increased exposure of terfenadine, as there's going to be no other enzymes available to metabolize the clearance of the active drug, and thus leading to the potential dangers of toxic exposure. So this is just an example of how a properly designed reaction phenotyping study can elucidate such a potentially fatal drug-drug interaction early on in the drug development process. Okay, moving on to the drug metabolizing enzymes themselves. There are two study types, and these two study types are drug inhibition and drug induction studies. And these are conducted to determine the perpetrator potential of a drug. Now, historically, there have been more victim drugs that have been withdrawn from market than perpetrators. So the first of these study types would be looking at the drug metabolizing enzymes, and is the drug an inhibitor of the CYP enzymes? So for a CYP inhibition study, human liver microsomes and a specific marker probe substrate of the various CYP isoforms are incubated either with or without a pre-incubation of the drug. And this is generally conducted at 7 concentrations of the drug. And this is used to facilitate the calculation of an IC50 value to determine if there's going to be any clinically relevant CYP inhibition. Now, I'm not going to go over this. I'm just putting it here for reference. But here is a list of the main CYP isozymes that are required to be investigated by the regulatory agencies, along with their preferred activity assays for those corresponding CYPs. So here is some data of CYP inhibition, and just 2 examples of CYP3A4 inhibitors, ketoconazole and mifepristone. Now, just to briefly orient you to these plots are the same, except for the drug that's being tested. The y-axis shows the activity of the CYP3A4 enzyme. And this is plotted as a function of the drug being tested on the x-axis. Either ketoconazole, which is on the left, or mifepristone, which is on the right. So ketoconazole, shown on the left, is an antifungal drug, and it's either pre-incubated with human liver microsomes prior to starting the reaction with marker substrate, or it was added directly with the marker substrate. Now again, CYP3A4 activity is plotted on the y-axis, and concentrations of ketoconazole are on the x. Now you can see here, the 2 curves are pretty much superimposable with each other. So there's no appreciable difference in the IC50 values in these treatments with and without a pre-incubation. So this is indicative that ketoconazole is a direct inhibitor of CYP3A4. And there's no shift in the IC50 curve that's observed between the 0-minute pre-incubation and the 30-minute pre-incubation treatments. However, shown on the right are the IC50 curves for mifepristone. So mifepristone, on the other hand, has a significant leftward shift in the plot for the treatment with a 30-minute pre-incubation. This shift in the plot of the IC50 curve is indicative that mifepristone is a time-dependent inhibitor. So as you can see, the IC50 goes from about 5.5 micromolar in the 0-minute pre-incubation treatment, all the way down to about 0.06 micromolar in the treatment with a 30-minute pre-incubation. So knowing if the drug it has direct inhibition versus time-dependent inhibition is important from a clinical standpoint, because time-dependent inhibition has a much greater drug-drug interaction risk. Now, this is because direct inhibition only persists while the parent is around, while time-dependent inhibition can last much longer. And in fact, in some cases, time-dependent inhibition requires the synthesis of new enzyme before that CYP will become functional again. And this is the case of mifepristone, and it was withdrawn from the market due to its potent and persistent inhibitory effect. This also represents an example of a perpetrator drug being withdrawn from the market due to a drug-drug interaction. Okay, the next in vitro ADME DDI study type related to looking at the drug metabolizing enzymes specifically would be CYP induction studies. Now, for CYP induction studies, human hepatocyte cultures are incubated with the drug, and messenger RNA or activity of the enzyme is measured. Now, the regulatory guidance does not specify which endpoint to use in CYP induction studies, but typically, messenger RNA is the preferred endpoint for induction studies. However, as I mentioned, the activity can also be assessed, and sometimes drug developers might choose to look at both to have a more robust data set. Now, the regulatory guidance requires cultures from a minimum of 3 donors be used, And this is to account for any inter-individual variability that might persist between the donors. And if positive induction results are obtained from any one of these cultures, the drug is considered as a positive inducer. And this is just the regulatory agencies taking the worst case scenario for the sake of safety of that drug. Now, just to briefly orient you to these three plots. So these three plots are the same. Each one is from a different donor. The y-axis is the effect or the fold change. So the fold change of the amount of messenger RNA for a given CYP. And again, it's functioned as a concentration, increasing concentration of the drug on the x-axis. So when the data are plotted this way and fitted with a curve, we're able to calculate an EC50, so a concentration of the drug that had a 50% max change in the max fold increase, and also an Emax. So what is the max fold change caused by that drug? So for both inhibition and induction studies, following up afterwards with these data can be done using either both basic or mechanistic models. And these can be employed to further determine if any of the results obtained might have the potential for a clinical drug-drug interaction and cause the requirement of a clinical drug-drug interaction study. Okay, moving on to the drug transporters. Now, these studies are conducted to evaluate if the potential of the drug to either be a substrate of cellular transporters or an inhibitor of these cellular transporters. Or to say another way, to assess if the compound has the potential to be a perpetrator or a victim of some of these drug-drug interactions. So why conduct drug transporter DDI studies? Well, drug transporters are responsible for the A, the D, and the E in ADME. Drug transporters play a role in the absorption, the distribution, tissue-specific drug targeting, and elimination of the drug. So drug-drug interactions can occur with different mechanisms. For one, the clearance of transporter substrates, or the victims, can be impacted by transporter inhibitors or inducers, which would be the perpetrators. And either one of these could either lead to toxicity or loss of efficacy of a drug. A real-world example of this are statins. So statins are taken up into the liver by hepatic uptake transporters, specifically OATPs. And in the liver, they function to reduce cholesterol. However, cyclosporin inhibits OATP transporters. So if someone is taking statins alongside cyclosporin, it could potentially lead to large increases in statin exposure. And this can lead to some toxic side effects, such as rhabdomyolysis, which is a breakdown of the skeletal muscle, and cells become released into the bloodstream, which can lead to kidney failure and possibly death. So shown here in these schematic representations are the various drug transporters that the regulatory agencies has listed as important to look at. I'm not going to go over this in too much detail, but I just put this here again for reference. So drug transporters are found in all cell types throughout the body. However, some drug transporters are more localized to certain tissue types and certain parts of those tissues. The transporters that are important from a drug development standpoint are the ones that are shown here in the boxes. This is also a good reference, and it shows not only the location of a lot of these various transporters that are important to look at, but also the direction that they are transporting their substrates into. So again, drug transporter studies can be done to either look at the substrate potential of the drug or the transporter inhibition potential of the drug. For these study designs, either polarized cell lines that are expressing a specific transporter are plated in transfected cells or vesicles. And for the assessment of substrate potential, the transport of the drug is evaluated with and without a known inhibitor to that transporter that's being evaluated. For inhibitor potential, the transport of a marker substrate is evaluated, and this is done in the presence and absence of the drug. So as shown here in this example, the apparent permeability and efflux ratios are plotted on the y-axis, and this is for inhibition on the plot that's shown on the right-hand side. So the apparent permeability is plotted on the y-axis, and the inhibitor concentration of the drug being tested is increased from the basal to apical side, and this is plotted on the x-axis. And again, the transport is also shown from the apical to basal side and the basal to apical side. So as you can see in this example, as the concentration of the inhibitor, the drug being tested, increases, the apparent permeability of the marker substrate decreases, or the efflux ratio also decreases. And this is indicative that in this case, this drug is an inhibitor of this transporter. So again, if you're interested in learning more about these drug transporter experiments in detail, we have some in great depth videos discussing them available on our website. So now that we've reviewed the different types of in vitro ADME studies, let's consider some examples of where taking time and the importance of designing a proper experiment and proper interpretation of the data really come into play. So I'm going to do this using a few case studies. Now, the first example here is of a CYP inhibition study design. And if you don't design the study properly, you can have the potential to end up with false negative results. So as shown in this example, clinically relevant, time-dependent CYP2C19 inhibition of omeprazole, or Prilosec, was missed for many years due to the use of a high protein concentration and longer incubation time in the experimental design. Again, these are CYP inhibition IC50 plots with the percent of control of the hydroxylation of marker substrate shown on the y-axis, again, as a function of increasing concentration of the drug, in this case, omeprazole, on the x-axis. So as seen on the left, when the study was conducted with 1 milligram of protein and 30-minute substrate incubation, similar IC50 values were observed. So here it was missed that omeprazole is a moderate time-dependent inhibitor. And this is based on the very slight shift that's seen in the IC50 curves. But when the experiment was conducted with a lower concentration of protein, in this case 0.1 milligrams per milliliter, as opposed to 1 milligram per milliliter, and a much shorter incubation time, 5 minutes versus 30 minutes, time-dependent inhibition by omeprazole was revealed. And this is shown as the four-fold shift in the IC50 value that's observed in the treatments. Now, why is this important? So as we saw, this information is important for the drug label. And time-dependent inhibition has a higher risk of drug-drug interaction than a direct inhibitor. So therefore, this is one example that's illustrating how designing the studies properly is very important when assessing in vitro drug-drug interactions. The next example is of a CYP induction study. So in a CYP induction study, and in the examples here in these plots, testosterone hydroxylation is plotted on the y-axis. And various cultures are ranked based on their testosterone hydroxylation activity on the x-axis. So after rifampin incubation, and rifampin is a known inducer of CYP3A4, the cultures are plotted again for their testosterone hydroxylation activity. So as you can see here, the cultures in the gray box have a very high induction, as shown that's shown in the third figure. Now these rank orders on the x-axis of these various cultures stay the same throughout these plots. Now, while these cultures at the very end that are highlighted in the box appear to have a very high amount of induction, their basal activity itself was very poor. And this is leading to supraphysiological increases due to the calculations that appear that the fold increase or the induction of these enzymes is several hundreds-fold. So these data indicate that these false results were because the cells were not fully confluent, and there may have been gaps in the culture. So the cells were not necessarily in the best of condition. So such data should be the ones that we are suspicious of. So good cultures will have high control CYP activity, thus lowering the magnitude of induction to something that is more physiologically possible, such as a 6 to 20-fold increase, and not this physiologically impossible increase of several hundreds-fold. So in this case, this is an example that shows when it comes to induction study results that more is not always better. And also that the data need to be interpreted in their entirety. And in fact, so when cultures are poor, there's a possibility that induction potential may even be missed entirely for your drug. Again, this example is underlying the importance of proper interpretation of your data and selection of proper cultures that have already been qualified for the assay that you're looking for. Now, the third example I'd like to give is of a reaction phenotyping study. Now, choosing the right test system to use in your study based on any information you have, or in this case, the structure and properties of the drug that's being investigated, is also of utmost importance. Now, as I mentioned in the beginning of this presentation, a lot of the sponsors are under the impression that these studies are simply box checking and insist that they only want to study the CYP panel of metabolites and the CYP metabolic pathways, as the guidance does state. However, all three guidances allude to the importance of assessing the potential metabolism of other drug metabolizing enzymes as well. So this example here is a great illustration of why just looking at CYP and just doing the box checking is not always the best course of action. So in this example, Ezetimibe can of course be metabolized by CYP3A4 when it's incubated with human liver microsomes and NADPH, or when it's incubated in recombinant CYP enzymes. But in reality and clinically, the results from this in vitro experiment are very misleading when based simply on the in vitro data and only using NADPH as the cofactor. So if you look at the structure of Ezetimibe, you'll see that there's a phenolic hydroxyl group, and therefore ezetimibe is readily glucuronidated even before the CYP metabolism can occur. So this leaves very little of the parent compound left available for CYP3A4 metabolism. So in this particular case, studying the role of UGT enzymes is more applicable than the CYP panel alone. Now, the point to remember here is that CYP metabolism is not the only enzymatic pathway. And depending on the structure of the compound, other enzymes might be involved as well. So this example stresses the importance of selecting the proper test system when designing studies and interpreting data for in vitro ADME DDI studies. Again, in this case, human liver microsomes would still be an appropriate test system. However, they should be fortified with UDPGA, the cofactor for UGT enzymes, instead. Or the use of human hepatocytes would have been a better test system choice, as human hepatocytes have all the metabolic pathways in operation. So now that we've reviewed the types of in vitro ADME studies and how to design the studies, let's look at the timing of the studies and where they should fall in the timeline of drug development. So this table here illustrates where in vitro DDI studies tend to be conducted based on various drug types. In this case, such as a typical small molecule, a small molecule drug with orphan or breakthrough status, and also larger drugs, such as biologics, including peptides, oligonucleotides, antibody drug conjugates, et cetera. So you can see that there's a lot of subjectivity when it comes to the timing of the studies. Now, usually it is advised to get as much data as possible, as early on as possible in the drug development program. However, we all know in the practical world, the priority depends on how much de-risking is required for each individual molecule, along with the drug developer's specific needs and what other work has previously already been done on the molecule. So as shown in the table here, for small molecules, it's generally preferred to look at the metabolism studies during lead optimization. CYP induction and inhibition, transporter inhibition, and substrate potential of some of the transporters should be done prior to IND filing. And reaction phenotyping and substrate potential for the remaining transporters could maybe be deferred until phase 1 to NDA timeframe. Now, for small molecules with orphan or breakthrough status, and peptides, oligonucleotides, or other biologics, some of these studies might be able to be deferred until later on, and perhaps run during phase 1 to NDA timeframe. So the most important point to be noted, though, is the drug developer should want to collect as much drug-drug interaction information on their drug as early as possible. Now, this most important reason is so you're not unnecessarily excluding patients in clinical trials who may be on different drugs, as that may lead to a delay in the approval process. And this is stated in the FDA guidance. A lot of, again, like I said, timing of the drug, these in vitro studies have to do with the specific drug developer, where they are in their program, the amount of risk they're willing to take, and the amount of de-risking that they want to do. So just in a nutshell, the timing really depends on the drug developer's risk tolerance and their de-risking strategy. So to conclude, why are in vitro studies important? It is because in addition to revealing the potential of any drug-drug interactions, they give an insight into future performance of in vivo studies. They assist in right test species selection for their tox studies, or will assist in first-in-human dose selection. They can help design, help guide the design of any clinical studies that are going to be used to follow up, or if there's even a need to follow up. In vitro ADME and DDI studies satisfy the regulatory interest, and they prevent any unnecessary delays in approval. As we saw from the areas of concern, the study design and interpretation of the data can be deceptively simple, yet they're actually very complex. And proper study design and data interpretation is extremely important to determine valid and reliable results. And both of these things can benefit from expert design and understanding. Timing of various in vitro DDI studies could depend on the drug class and the de-risking strategy that the drug developer has. And I'd also like to remind drug developers and sponsors to provide as much information as you can about your drug to the scientists that are going to be conducting these studies. You know, might get overlooked, but a lot of information that might be considered basic, such as molecular weight, the molecular structure, solubility, stability, these can be invaluable in helping to design the proper study in order to provide good results. And this information will go a long way to help the in vitro DDI scientist be able to guide you better when they have all the necessary information. And lastly, based on industry feedback that we've received, the majority of researchers have encountered holds or delays, have had to perform additional studies, or experience patient health issues or clinical failures that could have been avoided by conducting in vitro DDI studies early on. So yes, in vitro ADME DDI studies are crucial for a successful IND submission. I hope that this presentation has shown that the information provided from these studies early on to the drug development process has real utility, provides the drug developer with data to drive future decision making. It can save time and cost in the development process, and most importantly, helps to keep safety a priority for the patients who will ultimately be taking these medications. I'd like to thank you all very much for your time and for watching today. We have some time for Q&A, so I look forward to those. But I'd also like to say if there's anything we don't get to today, or if any questions pop up after the fact, or you have any further questions, please feel free to reach out to me directly or visit our website. And we have a lot of great scientific resources there, or you can contact us through the website as well. And that being said, again, thank you all, and I look forward to your questions.
Fantastic. Well, thank you, Andrew, for a great presentation. Andrew, you ready to answer a few questions here?
I am, and I saw some great questions come in.
Fantastic. Okay. All right. So first question here, Andrew. If in vitro DDI studies were conducted many years ago, for example, maybe before the 2020 guidance came out, would you recommend redoing such studies to ensure they meet the current guidance recommendations?
Yeah, and this is a great question we get asked often, and the answer is maybe. One would actually have to go in and look at the study design and see how the studies were conducted to make sure that they meet current guidance. If not, yes, we'd absolutely recommend to repeat those studies. But if they do meet current guidance, then there's probably no need to repeat them.
Fantastic, great answer. Next question here. So for CYP inhibition and induction conducted prior to the IND, we do not have information on clinical drug concentrations. So what concentrations would you recommend for these studies?
Yeah, another great question. And this is actually something that comes up a lot in our line of work, especially when clients are very early on in their drug discovery process for a particular compound. So the main concentrations that are, or the highest concentrations that are used, of course, are set forth by the regulatory guidance. But if that information, like say on dosing or Cmax, isn't known, we want to go as high as possible. And by going as high as possible, even if the Cmax isn't going to be that high, and say the regulatory agency wants to look at 50 times unbound Cmax, at least we're going high enough to cover our bases, or the drug developers covering their bases just in case. So in those situations too, we might recommend just going as high as even solubility or cytotoxicity allows, just to make sure that we're testing it high enough. So good question.
Yeah, it makes sense. All right, so if we see a borderline induction, do you think it would be necessary to conduct a clinical DDI study?
There's not an easy answer to that question. And my answer would be, it depends. There's a lot more that needs to be looking at. Perhaps no, you might not need to conduct a clinical DDI study if the clinical dose is going to be much lower than what we tested at. It would also be necessary to do the R equations, which are the static models put forth in the guidance, which could give some insight onto whether or not a clinical DDI study will be required, and also just to look at the data as a whole and determine if a clinical study is going to be needed as a follow-up.
Excellent, great answer. Another good one here. Is there a possibility that if we inhibited one of the CYP pathways, that other pathways might be automatically activated?
Yeah, these are all excellent questions. Yes, assuming that the compound is a substrate for other drug metabolizing enzymes, this could absolutely be the case. A great example for this that's well known is acetaminophen. Acetaminophen is easily metabolized by CYP2E1. However, inhibiting CYP2E1 can then doesn't necessarily alter its metabolism. It's then just metabolized by other CYP isoforms such as 3, 4, 1A2, etc. So other of the isoforms of a given family can step in to compensate for the inhibition of one of them. And it's also important to remember that it's not necessarily always in the same family too. Perhaps other enzyme families could take in, such as UGTs, other sorts of conjugating enzymes, things like that.
Yeah, really good point. Thanks for that. All right. So for CYP induction, do you usually perform a cytotoxicity assay? And if you were to perform it, would you use it from samples from the same well?
Yeah, so for CYP induction, the regulatory guidance does require that cytotoxicity be assessed during the assay. So it is our standard design at Xenotech that we look at cytotoxicity. We do this on a daily basis during our 3-day treatment of the cells. We do this for all 3 donors, and we also do it at all concentrations as well. So it's done in every single well, and those data are taken and compiled at the end of the experiment.
Excellent. Yeah, it looks like we actually have a handful of questions on CYP induction here. So for the same test, so how do you select the test concentration and how many of these do you pick?
Yeah, so as I answered before with the question on if, say, we don't know what our concentrations are going to be, the top concentrations to use for induction are picked or determined by what's set forth in the regulatory guidance. For example, the new ICH M12 guidance, the draft guidance that came out in May earlier this year, is pointing towards 15 times the unbound Cmax as the top concentration to use. When it comes to how many concentrations to pick, that's not necessarily put forth in the current FDA guidance. However, you have to pick enough concentrations to calculate an EC50 should induction be determined. So that's a minimum of 5 because you need at least 5 data points to make a decent curve. Our standard at Xenotech is to use 7 concentrations, and that way if we happen to see cytotoxicity at the highest concentration, we still have enough data points to be able to calculate the EC50 at Emax.
Great answer. I think in the interest of time, we'll just make this next question the last one. So again, for a CYP induction, do you get a mixed response? And how do you interpret that data? Does mRNA data supersede enzyme data?
Excellent question, and one that we get asked a lot because it is can be a common occurrence. So I'm not exactly sure what meant by mixed responses. That can mean different things. But according to the guidance, if even one of the 3 donors shows positive induction, then it's considered an in vitro inducer. But I think mixed in this question might also mean how do you interpret the data if you're looking at messenger RNA and enzyme activity? The guidance does not specify which endpoint to use. However, mRNA is the preferred endpoint. Enzyme activity data can also be added to have a more robust data set, and enzyme activity data can also be helpful if one is worried about suppression. There is always the case, though, when using enzyme activity that the drug or some of its metabolites might be an inhibitor of those enzymes, which could make interpreting the enzyme activity data more complex. And that's why messenger RNA is the preferred endpoint.
Fantastic. Yeah. So Andrew, yeah, thanks so much for the really fantastic presentation today. It's been a real pleasure having you with us.
No, thank you. And thanks for taking the time, Liam, and everybody else that attended.
Yeah, definitely. Big thanks to the audience here for participating. So in closing, we hope you enjoyed this scientist.com and Xenotech webinar, and we'll see you again next time. Have a great day, everyone.